great. I can start. Hey, so hi, everyone. I'm Yohan Yee. I'm a postdoc at McGill, and I'm honored to also be on the communications committee. So Chinwee's given us a great overview of how to make really cool-looking images. The goal of my tutorial is to actually show you practically how to do that. So the goal here is to make a simple image that looks like this on the right. So you kind of see these images in a lot of different publications where you have a statistics map overlaid on top of a brain. But what I want you to focus on is not so much the outcome, but the process of making this image. So what components go together, how they're overlaid on top of each other, how they connect with each other to make this. And I'll give you a walkthrough in all of that. If you have any questions at any point, feel free to either ask the question on the microphone or put it in the app, and Kevin, you can interrupt me at any time. And this is interactive, so if you want to follow along, if you want to code along, you can either go to this link here or scan the QR code. There is a Google Colab link, a Google Colab notebook, where you can run the code as we go through it, or you can access a static version of that through this web page. So everything I'm going to talk about is done in R, but it's programming language sort of independent, so there are other ways to do this. But if you are running things along in Google Colab, just make sure to change the runtime type and select R. So, there are a lot of different ways we can visualize brain images. There's a lot of different packages that help you do this. I've listed a few here. This is probably 10% of what exists, maybe less. And there are so many such that there are now papers that tell you how to visualize images, how to sort of put them together, what process to follow. In this tutorial, I'm going to focus on one specific type of image or one specific type of data, which is voxel-wise data, and how to plot slices of voxel-wise 3D brain data. When we think about that, I kind of think about plotting in terms of the spectrum, where you can use packages that are built specifically for plotting brain images. So it's very simple, but you're limited to specific plot types. But on the other end of the spectrum, you can create complex plots you can use sort of flexible approaches, but you really need to sort of understand the code that goes behind that. So an analogy I like to make is, we're all here in Korea. Chances are that most of us, coming from outside of Korea, don't really know Korean. We might know a few phrases that are very context-specific that'll help us get what we need at a restaurant or something, but really to communicate well, you need to understand a grammar. And so what I'm going to tell you about is what's called the grammar of graphics. So it's a framework to put together images. It's not a specific package, but there are different packages available in different programming languages that help you put together images through what we call the grammar of graphics. So in R, there's a package called ggplot. The equivalent in Python is called plot9. I'm sure there are others in other languages. I'm just not too familiar with those. In R, the grammar of graphics and ggplot really arose from Hadley Wickham's paper, A Layered Grammar of Graphics. And he writes, a grammar allows us to move beyond named graphics. So just as I said, beyond specific contexts, beyond scatter plots, beyond bar charts, we can start to really think about the deep structures that underlie these graphics and then kind of put them together to create anything you want. So what is this grammar of graphics? Well, Hadley Wickham proposes these different sort of elements of the grammar, things like data, mapping, geometry, statistics, scales, facets. They may seem a bit abstract, but I'll give you a bit of a walkthrough before we get into plotting brain images. So let's look at what these different elements of this grammar are. So on the right, I'm going to have some code for you, and on the left, I'll show you what that gives you. So we'll start off with some data. This data set, it's a very popular data set in data science, the iris data set. Essentially 150 observations of different flowers from the iris species. And what you have in there are 5 different columns or features Data on the sepal length, sepal width, petal width, and 3 different species of flower, 150 rows in total. So I'm trying to go to the next slide. Oh, okay. Okay, so in R, in ggplot, if you were to simply add data, iris, it would give you a blank plot. So this kind of makes sense, right? Like, so far we haven't provided any information on what to plot. You know, you have a lot of data here. You can make a histogram out of it. You can make a scatter plot out of it. There's many different things you can do. So what you need beyond this is the other element, one of the other elements of this grammar, which is called a mapping. So essentially what we're saying is we want to map one variable, in this case sepal length, to the x-axis, and the other variable on the y-axis, where we're going to map another column of this data set to that. When we run this, we see that now we have these axes, but nothing's actually plotted on that. What we need to do is add another element. So the next element that we're going to add to this plot is a geometry. So we're essentially saying we have our data, we've mapped different columns, so sepal length to the x-axis, sepal width to the y-axis, and we want to plot this data on these axes using a point geometry. So this is our third element. And essentially with these 3 elements, when you break it up into these 3 components, you get a simple scatter plot. The mapping can be anything, right? So instead of mapping just the x and the y-axes, we can also map a color onto a different column, or a column onto a color. So here I'm mapping color, I'm mapping species onto color, and that gives you your points that are plotted by the different species, or they're plotted in different colors based on the species. The other thing to note is that implicitly here, there's another element in this grammar of graphics that's being associated with this plot, and that's the scale element. So essentially, we're not just mapping a color or a species to color, but we're also putting a color bar to that. You can change the scale and say, okay, I don't want red, green, blue, but I want maybe yellow, purple, pink. So you can associate a different scale with that. But by default, there's a default color scale associated with this plot. And similarly, the x and the y-axes also have their own scale, right? So the scale essentially says, how do you take the sort of numbers that you have, and where do you plot them onto the screen for the axes? So you can sort of change that scale. You can use a log scale if you want to stretch the axes in different ways. You can use a root scale. There's different kinds of transformations and scales you can use. And just as another example, you can map species not to color, but to some other element, so shape, for example. Before we move on, I just want to get into a bit of the technicalities of ggplot and the sort of the layered grammar of graphics as it's implemented. So I've essentially moved the mapping into the geometry here. So before the mapping for shape was above here, it hasn't, in the plot, nothing's changed. And that's because before geom point was inheriting it from the original ggplot object. So you can customize different mappings to each geometry. And this will make sense as we go further. And finally, I kind of want to get into this last component of the grammar, which is called a facet. So that allows you to split the data into different groups and plot them separately. And so here we're splitting it by species. And oh yeah, one more component. So there's also this idea of a statistic. So before we simply plotted each point, each observation as a point, you can also summarize these observations using a statistic. So you can compute the mean from these different observations. You can compute histograms. And so if you use a different geometry, for example, a density geometry, a statistic associated with that is computed, and you get a plot related to that. And so what that helps with is, for example, if you wanted to do a linear regression and kind of show different trends in the data, you can use a geometry like geom_smooth that computes the statistic for you. It does the regression and it'll plot that on there. What you might notice is you have two different layers, two different geometries, and so essentially what you're getting is this layer effect where you're plotting geom_smooth on top of geom_point, and so you have your regression line on top of the points. So why am I telling you all of this? Well, this is what we're going to use in the tutorial to plot brain images. So just again, if you need the link, it's right there. And yeah, feel free to follow along and ask questions. So in the tutorial, we're going to work with a data frame that's called DF template. So you can view it if you're using R or RStudio by piping it into view. I'm just using this pipe notation, this functional programming notation. It's equivalent to simply running view of this data frame. And if you were to look at what the data frame looks like, we essentially have our image data, but we've put it into this weird table format where, we're explicitly writing out the positions of the voxels in world coordinates. So we have the x coordinate, the y coordinate, the z coordinate, and the associated intensity or the value of those voxels in this column that's denoted intensity, and some other metadata associated with all of this. So it might seem weird, but it's going to make sense because with the data in this format, you can now think about mapping the different columns here to the different aspects of your plot and getting brain slices in that manner. So for this tutorial, we'll make it a bit simpler. We're actually just going to take a single slice of this data. So essentially, we're going to take only, so this is a 3D image, and we're only going to take slices that correspond to the x value of 10. So in this case, because we're filtering it based on a single x value, that's going to give you sagittal slices. And so if you do that, essentially you get a single set of x values and then different y and z values, and those are your corresponding sagittal slice. So we can use the same process we used to plot, the points that we plotted before. We essentially have the same thing where we have our ggplot object, we feed it in the data, we feed in the mapping, we say, okay, on the x-axis we actually want to plot the y coordinate, the y world coordinate, and on the y-axis we want to plot the z coordinate, right? And so you get something by default that looks like this. We're using a geometry called geom_raster because our brain consists of, or our brain image consists of essentially equally spaced voxels or equally spaced pixels in this case, forming a raster image. And we're mapping the intensity column to the fill inside. So essentially it's filling in each box or each pixel based on the intensity value. So again, the same sort of grammar that we used to plot a scatter plot, we're plotting a slice of the brain. Doesn't look quite right yet, but what we can do is make certain modifications to this to kind of get this image to look right. So the first thing we can do is we can modify the coordinates, right? So we have physical units associated with the world coordinates. We're working with millimeters here. So your y coordinate is in millimeters, your z coordinate is in millimeters. We want to kind of hold them to sort of equal, or plot them equally. And so essentially we're fixing the ratio of the x and the y-axes on this plot to make the brain not squished. So that's one thing we can do. The next thing we can do is use a better scale, right? So as I mentioned, one of the elements of this grammar was a scale element, and we can explicitly add that scale element. We sort of say we want to modify the fill part of this, and we want to use a gradient. And so there are these different functions in ggplot that allow you to do this. So if you wanted to modify a color mapping, for example, you'd use scale underscore color underscore gradient. That would help you do that. And I've set the low value to be black, the high value to be white, so you get this grayscale image, and your coordinates are fixed as before. And if you kind of zoom in to the plot, you kind of see like your raster images. So really what ggplot is doing behind the scenes is it's plotting every single intensity value as a little box in this image. Each geometry has various different options that are specific to that. So geom raster allows you to interpolate. So we can kind of make our image look nicer by feeding it this interpolate equals true. The other point I kind of want to make is that, you know, we can use, in principle, any different geometry, right? So before we used raster, that made the most sense, but anything works. So here, for example, if we use geom point, you still get an image of the brain, but at the very core, you're plotting the individual elements as points. So let's go back to our raster image. We can increase the contrast to make things stand out a bit. So what we're going to do is add some limits. So we essentially say we're going to cap the low value and the high value and squeeze all the colors into that. And we'll remove the sort of guide bar that showed you the color values that was present on the right with this guide equals none option, because at the end of the day, those intensities are arbitrary. And we can now also add different labels. So from the previous talk, you saw that labeling axes were important. So we can really, with all the different options, start to customize every aspect of this plot. So here I've labeled this posterior to anterior, and the vertical axis is inferior to superior. And I've also switched a different theme here. So ggplot allows you to add different themes. You can customize your own theme. I added a different theme here to kind of change the background from gray to white with grid lines. Typically, if we just want to plot a slice, you know, we don't even necessarily care for that, so we can completely remove the axes. But if we do want to retain the axis labels or the axis titles, we can then add the titles back because this previous theme removes everything. So there are different sort of many different things you can do that allow you to really customize any image that you want to make with ggplot through these different theme options. So, you know, I switched the, so I removed everything first, added the axis titles back, changed the sizes of them, and then you kind of get an image that looks like this. So that's all great. So that's a single slice of the brain. But in practice, we typically want to show data that exists across the brain. So you have data across multiple slices. And so in the tutorial, you know, we'll go back to this original data frame that contains slices across different X values. So now you have multiple sagittal slices. And if you were to plot this directly, again, if I ran this, you'd get an image that looks exactly like this. Question?
Yeah. One question, just one question.
You can yell it out, I can repeat it.
Okay. Because you asked me to interrupt you, I'm interrupting you. So one question that just came in was, how do you get your nifty images, your brain images, into like a table like this? What's the best way to do that?
Great question. So several ways. I mean, one, you could do it manually. You have your voxel coordinates, your world coordinates, and essentially knowing the sort of extents of the images, you can read in all your data as a single, like 1D array and associate those individual coordinates with that. I'm actually going to show you a package that does that at the very end. If you scan the little QR code, there's a link to a GitHub with some R code that does that for you, so you don't have to, have to do it yourself. Right, so I was here, if I can go back. Perfect, thank you. Awesome, yeah, so if I were to run this code again, but with multiple slices in our data frame, you'd essentially get something that looks like this, right? So we don't see multiple slices, and that's because we have all our data in the same, the different slices, they exist within the same image. They're simply being plotted on top of each other with the very last slice being plotted on the very top, and you're essentially seeing the same thing again. If we were to rearrange the data, so we essentially reorder the different slices using this arrange command or this arrange function, now it plots the slice at the very other end on top. And so what you're essentially seeing are your multiple slices that exist in this data frame, but they're just being plotted on top of each other. And so, well, what do? You add a facet. So similar to how I showed you, where we split the flower, the iris data into the 3 different facets based on species. Here we're simply now splitting our brain data based on the slice world coordinate that we used. And simply by adding this, now you get your slices separated. There's other nifty tools that you can kind of use. So for example, the glue function is equivalent to Python's printf function, where it kind of, you can create your own string that is based on a variable. And so by adding it in, I'm just showing you that you can kind of modify the plot with code in place. And so rather than, just having the facet, the slice world coordinate being plotted at the top, you can kind of modify the panel headings up there. And so here you have x is equal to 20, for example, x is equal to 30. So it just gives more information that lets you know where the slice is. The other thing to know is that, there's a lot of extra space around these. Typically, we want to minimize any extraneous space. There's various different kinds of white space that is by default plotted. So there's white space around these different panels, and even within the panel, the raster image just takes up part of it, but there's also a little margin. And so, as I mentioned, everything is controllable, and so what you can do is you can remove that space within the panel by modifying your scales. So as I mentioned, the scale option allows you to transform the bounds of your data, and then you can remove that other white space around the panels themselves and the associated panel headings using the different theme options. So again, various different things you can do. Another thing you can add or you can play around with is the arrangement of the layout. So you can modify the number of columns you want to display the data with. So here I'm just using 2 columns, but in the end it's up to you. So that gives you what I call a slice series. Typically, once you have that, you want to overlay data on that, right? And the way we do that is simply use this feature of the layered grammar of graphics, where you just add different geometries on top of each other, and the data are rendered on top. And so all I'm doing, as compared to before, compared to this plot, is I'm just adding another layer. So the main component here is this geom raster layer. So this is our second component that's on top of our template data. This contains the data for the color map that you see. The color map comes from some component from a data decomposition of some data that we had, some volumetric imaging data. And we're, again, just mapping the intensity values onto that. And there's some other extra stuff around that. So we're saying we want a new different scale, so we don't want to use a grayscale. So we create a new scale, and we just define that scale below here. So we're using a different color. Dark red low, high values are denoted in yellow. And we're just saying anything outside of that this time, we want to make it transparent. So again, many different options to play with the data and modify it. The one thing I want to emphasize is that we are working with world coordinates, right? And so it doesn't matter what resolution your data is. So if you take data at a different resolution, you still have your data at the same coordinates. So if you were to just simply switch in a different data frame, the data would still be plotted in the correct place. And in terms of just switching in the data, I think that's one of the important things about the grammar of graphics is that because it keeps the data component independent from the other components like the mapping, the faceting, if you want to create a different plot with a different data set, all you need to do is simply just switch the data, right? So instead of plotting df component 1, for example, I can simply put in df component 2, which comes from a different image, and you get the exact same plot, but with a different statistics map overlaid on that. So that's really helpful for reproducibility if you have a specific kind of image you want to create, but you just want to switch a different data set in. This would help. So this is the two different components I showed you. So they were the two different color maps that existed in that plot. You can even combine them together to create a single plot, right? So the first table here on top corresponds to the statistics associated with what I call component 1. Second table is associated with component 2. I'm simply going to add a column for each row in table 1 called component 1, add a column in table 2 with every element called component 2, and we're going to join them together. So we're going to bind them by rows. And what this gives you is a new data frame that you can then just plot directly in there and add your component column as a facet. And so simply by making that one modification and adding another variable in your facet, now you've plotted both those components side by side. So you can have multiple components. If you have 6 components, you'd have 6 columns. But this is all done with just one additional layer or one additional piece of the grammar added to your code. So that gives you the slice series, but it's also often helpful to know where these slices exist in the brain. And so for that, what we create is called a locator plot. So it's the same thing again. I've just taken a coronal slice. This time I'm mapping the x coordinate to the x-axis here, but the code is the same as before. But with this grammar, because it's not brain specific, you can add any kinds of geometries and elements on top of this. So what we're doing here is we're adding a data frame called locator bar locations. And essentially, in the tutorial, if you have access to that, the data is there. We construct this data frame, but we essentially say this is where the slices come from. And by adding this geometry, we can plot the different locations of those slices. So we can, again, modify how it looks. We can modify the line type, the line width, and that gives you the plot of where the previous slices came from. And then you can also make certain modifications by filtering the data, removing the background around it. And by this simple addition, you completely remove the background. And finally, the last thing you can do is patch these things together. So there's a package in R called patchwork that allows you to simply define your first plot that I showed. I call it plot slices, the locator plot that we just previously did. You define a design matrix that shows you how you want to design the plot. So A corresponds to this plot, B corresponds to this, and you kind of create this design matrix and it'll join the plots in that manner. And patchwork does that here. So all of that was very simple, but I think the idea is that once you're able to construct these brain images, then you can start throwing on additional things. So here's just a whole bunch of different plots that we've created either for past papers or presentations or for upcoming papers. that kind of show you the kinds of different plots you can create using the grammar graphics, taking on different components. So for example, here we're showing the deformations in a mouse brain when we use deformation-based morphometry. And really all we're doing is just overlaying a geometry associated with a path that links together different points that allow you to create these graphics. If you're interested, the second part of this tutorial on that link that I showed with the QR code shows you how to make this plot here. So the atlas overlaid on the brain along with the labels on the thalamus. And if you want to see a presentation that's used this framework, have a look at Olivier Perron's presentation. He created some really cool images from a normative modeling study. And all the images that he's created come from this plotting framework. So with that, I'll take any questions. As I mentioned in the middle, if you want code to actually take your images and make it into this data frame, this long format data frame that you can use for the grammar graphics, I have a GitHub package called ggslicer, which again you can access by going to the link here or taking a picture of the QR code. And yeah, happy to take any more questions.
That's great. This is super helpful to see it walked through actually line by line to see how each edition of code changes something. So that was super helpful for me to see and hopefully others too. Please raise your hand or come up to the mic if you have any questions here for Yohan. One that came in on the app is about plotting slices versus plotting a surface. So I know things get complicated once you move to surfaces, but is there a system within this that you can use some of the same tools for surface plotting?
It's not easy. So that's a different beast. And the reason is that with surface plotting, you typically are also doing some ray tracing behind the scenes. Whereas here, we're simply just overlaying colors based on specific intensities. You can construct certain kinds of, surface plots. So for example, you can take a surface projection of the data by simply taking your data frame, summarizing the data based on sort of the top voxels on your surface and rendering that. But I think for any general plotting, it's a bit more difficult, which is why I think this is really, at the moment, best suited for voxel-wise data.
Another question that came in right at the end of Xinhui's question and answer portion, but that I think both of you could perhaps give your thoughts on is how do you decide on the tools that you use for plotting? So I know you've sort of committed into this workflow, but at what point did you decide what either software package or graphical tool to use? Do you change it? How do you think through the logistics of making that?
Sure. Honestly, I think I would just say use whatever's the easiest. It's kind of like the analogy, if people ask like photographers, what's the best camera? The answer that they usually say is the one that you have with you at that time, right? And I would say it's the same thing. If you're familiar with some framework or some package and that gives you what you want, by all means use that. But if you want something more flexible, something that might allow you to do a bit more than any individual package, then I would urge you to consider this something like the grammar of graphics.
Yeah, I want to add that actually Yohan just showed a figure at the beginning how to choose the tool for visualization. I think it starts with the programming language. You can choose the programming language that you are mostly familiar with, and then you can also choose a tool based on the type of visualization and also your data type, and then that will help you to define which tool you want to use.
Any other questions? Okay. Thank you, Yohan.